My name is Hannes Link. I'm a professor for microbiology at the University of Tübingen, and my group is interested in bacterial metabolism. And we think bacterial metabolism is so interesting because bacteria can do something very special. They can utilize simple nutrients like sugars, and from these nutrients they produce all the building blocks that they need to grow to produce a new bacterial cell. And we think this flexibility and this efficiency of metabolism is very interesting, and if we understand it, there are a wide range of applications. So understanding metabolism comes with its own challenges, because in order to utilize nutrients and make all the building blocks that bacteria need to grow, they have to run hundreds of biochemical reactions all at the same time, simultaneously, and these reactions are very quick. And these reactions are organized into so-called metabolic networks. These are pathways or modules. Each module has its own function, and also the modules are connected, so that at the end you have a very complex network of these reactions. And the good thing is that today we have a very good understanding of these metabolic networks, at least about the structure and the topology of these networks. This means we know exactly which reactions are in the network, which metabolites are involved, and which enzymes. The problem is we don't understand the control of these networks, meaning what sets the speed of each individual reaction. Now, this might be a bit complex, so to make a simple analogy, you can think of a factory where the starting material are these nutrients, these sugars, for example, and then they are converted into the final product, the bacterial cell. In order to do this, there are hundreds of individual steps. And again, we know each step in this factory very well, where it is, what it is doing, but we don't know what sets the speed of this reaction. And this is really a complex task, because if only one of these 100 individual steps is not functioning very well, then the final product cannot be produced. So what are the applications? Now, understanding control of metabolism or function of metabolism has various applications. One application for us is synthetic biology or metabolic engineering. Here we want to reprogram these metabolic networks such that they don't produce a new bacterial cell, but rather a chemical that is useful for us. This could be bioplastics, biofuels, or pharmaceutical products. Then another application is antibiotic research, because many antibiotics, they are targeting metabolism directly, or at least reactions that are associated to metabolism, like protein synthesis, for example. And now in this case, we want to create a situation that I just explained, where we want to block as many of these individual production steps as possible, so that the final product cannot be made, that a pathogen in this case cannot grow, and in the best case that it dies. Yeah, and maybe one of the most exciting applications is microbiome research, because we have many bacteria living together, meaning we have many metabolic factories, and from all that we know today, they are not working in isolation. We can assume that they are highly interacting, meaning some of these metabolic factories are cooperating, they're exchanging certain products, but other factories are in competition, for example, for a certain resource or a certain nutrient that they both need in order to make their final product. So the methods that we are using focus on two key technologies. One technology is CRISPR, where we can perturb and engineer metabolic networks to change their function. And the second technology is mass spectrometry, specifically metabolomics methods, where we can then measure the metabolites and the fluxes in these networks, kind of to see what happens if we engineer the network, what are the consequences. And another important approach is multi-omics approaches. So we do metabolomics in our own group and also some transcriptomics, and then with collaborators we can also measure the proteome of a cell. And here really a key challenge is now to integrate these different data types and understand, for example, how metabolism or the metabolites at one end influence expression of certain genes on the other end. And here we're trying to understand how metabolic networks are now interacting with other networks in the cell, like genetic networks where gene expression is changing. So now in microbiome research, we can really benefit from the research environment in the Cluster of Excellence, where we have experts in microbiome research. So we get a lot of bacteria, and then we can make synthetic communities of these bacteria. These are bacteria from the gut, but we also study the nasal microbiome, and then we just grow a few of these bacteria together and then try to understand how the composition of these very small communities influences the metabolism of the individual bacteria that are growing in these cultures. So we're asking, how does the microbiome composition influence metabolism? And we're also asking, can we somehow infer from the metabolic signature of these bacteria a certain function? And the function we are studying at the moment is colonization resistance against a pathogen, Staph aureus in this case. One simple example, what we did is we used now CRISPR to perturb each chemical reaction in the metabolic network of E. coli. These are around 1,500 reactions. We perturbed each reaction individually and then studied the consequences with our metabolomics methods. And one thing that we learned is that very often E. coli has very specific mechanisms to compensate or to buffer these perturbations. For example, in some cases, you have a bypass, just go around the perturbation that you have in your network and compensate this. Now, in antibiotic research, we are interested how these perturbations in the metabolic network now affect the response of E. coli, in this case, to antibiotics. And here we learned that there seems to be an interaction between the metabolic state of E. coli and the way how it responds to an antibiotic. And this seems to be very specific effects. If you have a perturbation in one pathway, it helps you against one specific drug. If you have a perturbation in another pathway, it helps you to deal with another drug. And here we learned that really the metabolic state seems to play an important role how antibiotics work. So in synthetic biology, we try to engineer control of metabolism. So we try to engineer control elements. And what we learned that temperature-sensitive proteins are actually very effective control elements because we can very easily control temperature in the lab, but we can also control temperature in industrial settings, for example, in a bioreactor. What does that mean? So what could be now really a new therapeutic concept? And this could be that we take a clinical isolate, so a patient has an infection with a specific strain, and what we can do nowadays very easily to sequence the genome of that strain, so we know exactly which mutations are in these strains compared to a reference genome, for example, but we still don't know what is the function of these mutations. And now we can envision predictive models. This can be mechanistic models, but also more data-driven, machine learning guided models that predict now what is the function of these mutations, and we can then go one step further and now predict like a personalized treatment, what is the best antibiotic, what is a good combination theory, and maybe what even is a good diet to take with a certain antibiotic. What is the outlook? What's the vision of our research? So we are beginning to understand how a single mutation in the genome that we introduce now artificially with CRISPR, how this mutation translates into an effect in the metabolic network. And now one vision is now to really design this at the computer in a synthetic biology project, for example. What are the genetic changes that are needed to acquire a certain function? But at the same time, also a future vision is now to use this understanding and understand how a clinical isolate from bacterial isolate from the clinic, which also comes with many little mutations, how these little mutations now affect metabolism of such a clinical strain, of a pathogenic strain, which can cause disease. And then propose something like the personalized medicine. What is the best treatment against this specific bacterial strain?